Hi, thanks a lot for having me. It's really a pleasure to be here virtually. My name is Joe Pickrell. I'm the co-founder and CEO of a company called Gencove, and the title of my talk today is Low-Pass Sequencing Increases the Power of Genome-Wide Association Studies and Decreases Measurement Error Polygenic Risk Scores Compared to Genotyping Arrays. And just as a bit of background about the motivation of the company and the technology that we work on, what we've seen is that there's this growing need for acquiring and interpreting quality genomic information in a cost-effective manner in a number of different segments of genomics, from consumer and population genomics to agriculture and R&D. And just a couple examples here. On the left-hand side, I'm showing the number of consumer genetic tests done per year, reaching a peak of about 8 million probably in 2018. And on the right, I'm showing the number of genotyped cattle. This is actually the number of genotyped dairy cattle in the United States over the past decade or so, which went from 0 animals genotyped per year in 2008 up to 500,000 or more now in 2017 and these days. And so for applications like these, where there's that operate at fairly large scale, really the challenge is what's the optimal way to measure a genome given a particular scientific or commercial problem at hand and practical financial constraints. And historically, this has been sort of a balancing act between two different types of technologies. On the one hand, you have deep whole genome sequencing, which has been decreasing in price, but is still a ballpark of $1,000 a genome. And on the other hand, for these really high throughput applications, the only really practical genome-wide technology was a genotyping array, where you measured a preset number of markers, maybe tens of thousands or hundreds of thousands or maybe a couple million genetic markers that you know are segregating in a population, and you use that as an approximation of the entire genome. And so what we've been working on is technologies around what's called low-pass whole genome sequencing that outperforms arrays for these particular types of applications. And so what is low-pass whole genome sequencing? It involves a few different pieces here. So a small amount of sequencing on a target individual. And when I say low-pass whole genome, I'm talking about 0.4x to 1x, maybe 2x or 3x sequencing, or sometimes as low as 0.1x, but ballpark around half an x to 1x sequencing coverage of an individual, followed by analytics, sort of imputation to large haplotype reference panels, and then the output of that, a small amount of DNA sequence on an individual plus large haplotype reference panels and software is the output of high-quality variant calls and additional downstream analytics. And so really what's made this possible over the past few years is a couple of things. Upstream at the sample preparation type level, there are now really high throughput and cost-effective ways of taking a little bit of DNA and turning it into a sequenceable library. There's the drops in sequencing costs. So, of course, that slide that I showed on the previous slide of the drops in sequencing cost plays a role here. And so you can go from a DNA sample to a small amount of sequence data in a relatively inexpensive manner. And then there's the advances in the analytics, so the large haplotype reference panels from different species and the algorithms to use those to create high-quality variant calls and additional downstream deliverables. And so where we come in is generally on the analytics side. So we developed a suite of software tools that enable applications that were previously not possible with low-pass whole genome sequence data. High-quality variant calls, that's sort of the table stakes here, and then additional types of deliverables like ancestry or parentage analysis, disease risk, and trait scores. And we provide a software as a service platform that ingests sequence data and outputs all of these types of deliverables in an easy-to-use manner across an array of species. Though today I'm going to be talking about human genetics primarily. And so what I wanted to talk about in this talk is some of the validation that this actually works. So how does imputation accuracy from low-pass sequencing compare to that from genotyping arrays? We did a study. This was funded by the NIH in the United States. We took 60 European ancestry individuals and 60 African ancestry individuals from the 1,000 Genomes panel. We sequenced them in triplicate at target coverages of 0.5x and 1x coverage on Illumina sequencers, and a subset of these, 30 European and 30 African individuals on the BGI-Seq 500 at a target of 1x coverage. And we also genotyped these individuals in triplicate on the Illumina Global Screening Array at about 600,000 single nucleotide polymorphisms. And so what we can do is, since these individuals have deep whole genome sequence, is we can impute them sort of in a leave one out design. So take a little bit of sequence data, impute it against the haplotype reference panel, and compare to the quote unquote truth, which we have from the deep sequence data. And we can do the exact same thing off of the array data. And so then we can compare. So we do the small amount of sequencing data, we impute up to the whole genome, and compare to the truth whole genome, and we do the same from the array. We take the array data, we impute up to the whole genome, and we compare to the truth data. And then we can ask which performs better and how do they perform. And so overall, we see that low-pass sequencing data has a higher imputation accuracy compared to this particular genotyping array. In each of these panels here, the x-axis is the allele frequency of variants in the population, and so each point is an average across a bin of SNPs of the same allele frequency. And on the y-axis is the non-reference concordance, and so this is it's like concordance, but restricting to non-reference alleles to avoid any particular types of reference bias. And so in the left panel is this metric for African populations, and the y-axis is this metric for European populations, and since it's a concordance metrics, higher is better. And so this metric, non-reference concordance to the quote-unquote truth high coverage sequencing data. And so what you can see is in the light blue there is the array. It performs the least well out of all of these different technologies. In dark blue is the Illumina sequencing data at a target of 0.5x coverage. In orange is the Illumina sequencing data at a target of 1x coverage. And in red is the BGI sequencing data at a target of 1x coverage. And so there are a bit of differences between the sequencing platforms here, but this is primarily driven by the library prep methods that were used for these different sequencers. And I can get into that later if it's of interest. And so non-reference concordance is a useful metric, but if you're running, for example, a genome-wide association study, the metric that you care about is imputation R-squared. So if you take an individual single nucleotide polymorphism and you compare it to the truth across a number of individuals, you can compute a correlation of the imputed dosages of the alternate allele to the truth. And that metric is directly correlated to the power to identify an association in the genome-wide association study. So if you were to run an association study looking for disease risk variants, higher imputation R-squared means higher power to actually detect those variants, those causal variants in the association study. And so this is a similar style of plot to the last one. On the x-axis is the minor allele frequency, and on the y-axis is this imputation R-squared metric, calculated again as an average across all SNPs in an allele frequency bin. And again, on the left is the African population, and on the right is the European population. What you see is qualitatively similar results. The array performs the least well, followed by the 0.5x sequencing, followed by the 1x sequencing. And if you put numbers on this, effectively what you've done is you've increased, just by switching technologies from this genotyping array to low-pass whole genome sequencing, you've increased the power of a genome-wide association study in an African population by something like 10 to 15%, in a European population by something more like 5 to 10%. And that's simply by switching from an array to low-pass whole genome sequencing. And so that's the sort of the key metrics we look at just for variant calling performance. But as you all know, the high quality variant calling enables downstream applications in things like disease risk prediction. And so we were also interested in how this performs for polygenic risk scores. As you know, polygenic risk scores are these metrics that combine the effects of thousands or tens of thousands or millions of SNPs across the genome to predict an individual's risk of disease. And so what we did was we computed two clinically important polygenic risk scores, one for breast cancer and one for heart disease on all of these individuals. And we're taking the weights for these polygenic scores from the literature. So these are pre-computed weights. These are existing validated polygenic scores. And what we're asking is, if we apply these scores to these different data sets, how accurately do we actually estimate the score? And so we quantify this with a squared error. So we have the truth data again, and so we compute the polygenic score off of the truth data, and then we can compare that to the polygenic score computed off of these different technologies. And in each of these squares here are the different populations and disease risk estimates. On the x-axis is the coverage, the sequencing coverage of an individual, and on the y-axis is the squared error. And so these different experiments here, experiment A was the half-x sequencing of Illumina data, B was the half-x 1x sequencing from Illumina data, C I'm not going to get into, and D was 1x sequencing of BGI data. And what you can see is that the higher the coverage here, the lower the squared error. And the dotted line there is the error off of the arrays. And so you can see that as you move to higher and higher coverage, the squared error decreases and decreases to the point where it's on average below that of the arrays at about half an x or 1x coverage. And so across both of the populations and diseases, the measured polygenic scores estimated from low-pass sequencing data were more accurate than those estimated from the arrays. There's one exception there. In the top right-hand corner, the breast cancer score computed in European individuals, it's only at a 1x coverage or so that it matches the performance of the array. But in all of the other situations, the low-pass sequencing data increases the accuracy of the polygenic risk score measurement compared to the array. And so what our platform does, the Gencove software platform, takes in FASTQ files off of a sequencer and performs automated quality control, genotype imputation, polygenic risk score profiling, ancestry analysis, and a number of other sorts of deliverables. This is just a screenshot of how this looks. And so this is a scalable and HIPAA-compliant platform running on Amazon Web Services, and happy to talk more about that afterwards if of interest. But so all of these results can now be delivered in sort of a high throughput, scalable way. The last thing I wanted to mention is that we've tested the performance of low-pass sequencing across different DNB-seq sequencers, coverage levels, and inputs. And this is just one last slide here. Working with the team here, we got data from the DNB-seq G400, the T7, and the T10 on a single reference sample. This is the famous NA12878 at different amounts of input DNA. And at different depths of 0.6x, 1x, and 4x. And so we computed these same sorts of metrics on data generated by these different sequencers. And overall, we obtain high-quality low-pass sequencing data across all of the different DNB-seq machines and DNA input amounts. So in each of these squares here, the x-axis is the sequencing coverage, and the y-axis is the non-reference concordance. And so what you can see is basically uniformly across all of the sequencing machines that the lower coverage, 0.6x coverage, yields an average non-reference concordance of about 94% and goes up to 97% at 4x coverage. And just to benchmark these numbers as a comparison, the average non-reference concordance from imputed global screening array data is about 90%, so well below even the lowest coverage here. And so just to wrap up then, what I've shown is that low-pass sequencing increases power relative to genotyping arrays for genome-wide association studies, particularly in non-European populations, and that it increases the accuracy of polygenic risk scores. I've showed you a bit about the Gencove platform, which allows this fully automated workflows from FASTQ to imputed variant calls and polygenic risk score calculation. And really this kind of scalable analysis combined with sequencing hardware allows any lab to reach truly genome center levels of throughput. Finally, I'll just point out a couple references here. We've posted a lot of the results here if you want more detail on bioRxiv on the paper on the left. And there's additional DNB-seq data in collaboration with the group at MyHeritage in the preprint on the right, which is also available on bioRxiv. And with that, I'll thank everyone from Gencove and BGI who is involved in this work, and I'm happy to take any questions. Thank you.